Spatial vision. Many basic phenomena in spatial vision can be linked directly to the characteristics of the neural structures that we have been discussing. We know, for example, that vision at high light levels is mediated by cones, while vision at low light levels is mediated by rods. Again, vision at high light levels is mediated by cones, while vision at low light levels is mediated by rods. Fundamental aspects of vision at different light levels can be related to the properties of the cone and rod photoreceptor systems. We began by discussing some of these fundamental perceptual phenomena, which provide a platform in the perceptual domain on which we can build later discussions of more complex phenomena in spatial vision. Again, we're going to start out by talking about fundamental functions in photopic and scotopic vision. Scotopic vision is the vision at the low ambient light levels, typical of nighttime, mediated by the rods. Then we have what's called mesopic vision, vision at ambient light levels, intermediate, and again, meso meaning between or in the middle, between phototopic and scotopic levels, typically of dusk, mediated by both rods and cones. And then we have photopic vision, which is vision at the high ambient light levels, typical of daylight or daytime, mediated by cone photoreceptors. So you're going to want to remember that the scotopic vision is vision at low light levels, and it is using mainly rods. Mesopic vision is ambient light between light and dark, and it uses both a mixture of rods and cones. And of course, then we have photopic vision, vision at high light levels during the day, typically mediated by cone photoreceptors. And of course, then we might want to often think of other differences between rod and cone systems that might affect driving performance, for example. We also have what's called luminance grading, which is a laboratory stimulus used in order to study spatial vision. It is a striped pattern containing alternating light and dark bars, commonly with a sine wave luminance profile. And then, of course, there are stimulus dimensions of luminance gratings. The two left-handed gratings, for example, we could see different grating patterns in contrast to different luminance gratings. Some differ in orientation. We could have bars that go left and right, ones that go up and down. We have horizontal, vertical, diagonal. We have what's called spatial frequency, which is a measure of the fineness of a grating's bars in terms of the number of grating cycles, for example, bright-dark bar pairs per degree of visual angle. Psychophysical experiments often use sinusoidal gratings in which luminance varies smoothly from bright to dark and back again with a profile that conforms to a mathematical sine function. Since the advent of computer-controlled psychophysical techniques, the preferred method of measuring thresholds is the method of constant stimuli. And we have what's called contrast threshold, which is contrast threshold values are often converted into contrast sensitivity values by calculating the reciprocal of contrast threshold. For example, you have 1 for every contrast threshold. A threshold contrast of 0.05 becomes a contrast sensitivity of 20, and a threshold of 0.02 or 0.2 becomes 5. The right hand, for example, graph plots data in the left-hand graph in terms of contrast sensitivity. The plots are called contrast sensitivity functions, or CSFs, and spatial contrast threshold, or SCT, is the minimum contrast between the lightest and darkest parts of a pattern required for it to be reliably detected by an observer. Lower values indicate better performance. Spatial contrast sensitivity, on the other hand, is the reciprocal of spatial contrast threshold, where higher values indicate better performance. Then we have what we mentioned as CSF, which is contrast sensitivity function, which is a graph of spatial contrast sensitivity to luminance gratings, plotting sensitivity as a function of grating spatial frequency. Then what's called contrast ratio is the ratio between the amount of contrast in an optical image and the contrast in the original stimulus, and the values near unity indicate the near perfect transference. And then OTF is the optical transfer function, a graph of an optical system's ability to transfer luminance gratings, plotting contrast ratio as a function of grating spatial frequency. And again, we have spatial frequency and CPD and contrast threshold, as well as the contrast ratio. Then we have other key terms in terms of temporal contrast sensitivity, the reciprocal of the amount of contrast between the brightest and the darkest phases of a flickering stimulus that is required for a subject to detect the flicker. We have also temporal frequency, which is the alternation rate of a flickering stimulus measured in hertz for the number of flickering cycles, bright-dark alternations per second. And temporal contrast sensitivity function, which is a graph of temporal contrast sensitivity, plotting sensitivity as a function of flicker temporal frequency. And so we have the contrast sensitivity on the Y value and the temporal frequency measured in hertz on the X value part. And then we have what's called spatiotemporal contrast sensitivity, which is sensitivity to combinations of spatial modulation and temporal modulation, namely luminance gratings with flickering bars. Space-time plot is a plot that represents luminance variation over both space and time, with space plotted horizontally and time plotted vertically. And of course, we have origins of variation in the spatiotemporal sensitivity. For example, neural factors influence the shape of the spatial and temporal CFSs, or CSFs, CSF, which, if you remember the CSF, it remains contrast sensitivity function. Again, contrast sensitivity function, or CSF, is a graph of spatial contrast sensitivity to luminance gratings, plotting sensitivity as a function of grating spatial frequency. Again, we're talking about there are two alternative accounts of how the properties of center surround receptive fields can explain the spatiotemporal S or CSF. One account is based on changes in the receptive field organization of a single cell population, and the other is based on differing responses of two distinct populations of center surround cells. Again, we have what's called receptive field organization, and of course we have spatiotemporal CSF, which is a graph of spatiotemporal contrast sensitivity, plotting sensitivity as a function of grating spatial frequency and temporal frequency. And then, of course, we have a spatial scale. The sustained channel is a channel of processing in the visual system that is most sensitive at high spatial frequencies and low temporal frequencies. A transient channel is a channel of processing in the visual system that is most sensitive at low spatial frequencies and high temporal frequencies. So a transient channel is the opposite of a sustained channel. Although both of them are channels of processing in the visual system, the sustained channel is most sensitive at high spatial frequencies and low temporal frequencies, while the transient channel is most sensitive at low spatial frequencies and high temporal frequencies. Then we have different kinds of theories, but I'm just, there's also spatial filtering and Fourier theory, psychophysical evidence for multiple spatial filters. There's adaptation, what's called threshold measurements, such as a comparison of contrast thresholds before and after the period of adaptation reveals what's called threshold elevation, which is the contrast threshold for a test stimulus is higher following an exposure to an adapting stimulus than before adaptation. Adaptation is one of the most widely used techniques in visual psychophysics. The basic paradigm involves 3 stages: pre-adaptation to psychophysical measurement, the adaptation to an inducing stimulus, and then the post-adaptation psychophysical measurement. A comparison between the pre-adaptation and post-adaptation measurements reveals the effect of the adapting stimulus on the performance. Measurements involve either threshold or supra-threshold stimuli. And we have supra-threshold measurements as well as threshold measurements. Size after effect is a change in the apparent spatial frequency of a test grading following exposure to an adapting grading. And then tilt after effect, which is a change in the apparent tilt of a test stimulus following an exposure to an adapting stimulus. And then there's what's a term called masking. Masking is a rise in the contrast threshold for a test stimulus in the presence of a second masking stimulus. We can assume that masking is effective because it corrupts the response of filters tuned to the test spatial frequency. It injects activity that is not correlated with the presence of a signal. And vernier acuity, which is an observer's acuity for detecting the direction of small spatial offsets between collinear lines or edges. And of course, we can see the intensity of edges and images are defined by steep changes in luminance across space. And then we have what's called hyperacuity. Hyperacuity is acuity performance in which the observer can detect changes in spatial location that are smaller than the distance between adjacent retinal photoreceptors. And again, we're talking about processing stages involved in current theories of edge localization in the visual system. A standard psychophysical test of edge localization performance involves what's called vernier alignment. We're talking about feature localization and the importance of edges and uses of spatial filters and theories of edge localization. Of course, peak is a point where the value of a function such as a receptive field response reaches its maximum. A primal sketch would be a relatively primitive representation of local spatial structure in an image produced by the initial stages of visual processing. Then we have some other terminologies as well, some other terms such as visual texture, repetitive variation in local luminance, contrast, size, and orientation in order to create an extended pattern. And the filter rectify filter, which is FRF, which is a sequence of image processing operations thought to mediate texture analysis. Its output discriminates between image regions that are differing in visual texture. And of course, then it goes over some brief psychophysical evidence on texture edge localization, and then they speak about texture analysis and what's called scene gist. And then of course, spatial frequency selective receptive fields lie at the heart of stereoscopic depth and motion computations. Concerning fundamental functions, the shift from cone vision to rod vision, or dark adaptation, takes approximately 30 minutes. Dark adaptation is accompanied by a shift in the relative brightness of surfaces that reflect different wavelengths due to the differing spectral sensitivities of rods and cones. And of course, it takes the eyes a little bit of time to adjust to the light from going in a light space to a dark space, or vice versa. The spatial contrast sensitivity function shows that human observers are most sensitive to gratings of medium spatial frequency. Sensitivity to spatial frequency declines rapidly at higher frequencies and gradually at lower frequencies. The temporal contrast sensitivity function shows maximum sensitivity to medium flicker rates at 8 Hz. Sensitivity to temporal frequency declines rapidly at higher frequencies and more gradually at lower frequencies. Spatiotemporal contrast sensitivity depends jointly on spatial frequency and temporal frequency. Sensitivity to spatial frequency is bandpass at low temporal frequencies and low pass at high temporal frequencies. The shape of the spatiotemporal contrast sensitivity function can be best explained by the properties of Parvo and Magno cells in the visual pathway. Concerning representation at multiple spatial scales, natural images contain information at many different spatial scales or spatial frequencies. Fourier theory provides a method of decomposing complex images into their spatial frequency components. Cells in the visual system can be viewed as spatial frequency filters, tuned to respond to only a subset of spatial frequencies. Psychophysical evidence for multiple spatials or spatial filters in the human visual system comes from contrast adaptation and from masking experiments. Concerning uses of spatial filters, spatial frequency-tuned filters in the human visual system are used in several computations for feature localization, for texture analysis, scene gist analysis, and in the early stages of stereo and motion processing. Theories of luminance edge localization combine the outputs of multiple spatial filters in order to infer the presence of features such as edges and lines. Some theories extract features before combining information at different spatial scales. Others extract features after combining the outputs of filters at different spatial scales. Theories of texture analysis include additional intermediate processing stages in which a nonlinear transform is applied to the filter outputs before second stage low spatial frequency filtering and feature extraction. Experimental evidence indicates that the rapid processing of scene gist is based on the pattern of response in spatial frequency and orientation tuned cortical filters. And of course, Fourier analysis can also be applied to vision. It was first used to study auditory waveforms and the representation in the auditory system, but Fourier analysis can be widely applied and used in the study of vision as well. Again, when we're studying 1D spatial images, 2D spatial images, as 3D human beings. Next, we'll talk about shape and object perception.